This screencast is over immunohistochemistry, given by Katie, Brianna, Nolan, and Kaylee.
Immunohistochemistry, or IHC, is a technique that is used to examine protein expression and localization in tissue morphology. Basically, the immunohistochemistry technique is used whenever scientists or researchers are looking for a disease cell, protein, or enzyme. There are 3 linked IHC methods, but the focus for this screencast is the immunofluorescence method. It is not the fastest process, but is extremely efficient and known to be the best out of all of the IHC methods. This technique works by using specific antibodies, which are response proteins produced by the body designed to target specific antigens or foreign substances that induce immune responses and are found on cell and tissue surfaces. IHC is helpful in the diagnosis of diseases such as cancer. It may also be used to help tell the difference between different types of cancer. Fluorescent probes are used to detect protein location and activation, identify protein complex formation, and conformational changes, and monitor biological processes in vivo.
The first step is to collect a tissue sample, which can include tissues, embryos, or single cells. In order to prevent breakdown of cellular protein and degradation, rapid preservation is required. Next, the tissue must be sliced and fixed to a slide. This is accomplished by using a microtome, or blade, used to cut thin sections from the tissue sample. The sample is mounted to a glass slide with tissue adhesive and dried. The slide is then quenched and buffers are added to block nonspecific sites where the primary and secondary antibodies may bind. The primary antibody is applied to the slide, which binds to the target antigen on the tissue surface. The secondary antibody is applied to the slide, which binds to the primary antibody and contains a conjugated fluorochrome. The resulting slide uses fluorescent light to detect target antigens. This is due to the conjugated fluorochrome attached to the secondary antibody. In this technique overview, the IHC method will be described with what is happening at the molecular level. A cell has specific antigens that target antibodies bind to. The primary antibody is applied to the sample, which binds to the antigen on the cell. A secondary antibody with a fluorochrome attached is added, which binds to the primary antibody. After the application of both antibodies, the sample is subjected to a light source to provide fluorescent excitation for color imaging.
Other common methods used for protein detection include ELISA, Western blotting, and immunoprecipitation. While these techniques are valid to use for protein detection, IHC poses advantages over them for the following reasons. IHC involves specific antigen to antibody reactions. This is advantageous over traditional special enzyme staining techniques because it can be used to identify numerous proteins, enzymes, and tissue structures, as well as spatial location. Also, detection rates with immunofluorescence histochemistry have higher detection rates and higher sensitivity in results, providing better images for diagnosis. IHC can be utilized for a variety of clinical situations. Infections such as Hep B and Hep C can be validated by visual expression of the affected tissue. Brain trauma can be identified by varying stains to detect axonal injury. IHC has helped with subclassification of neurodegenerative disorders and the development of criteria for their diagnosis. Muscular diseases such as muscular dystrophy can be diagnosed by looking for specific protein abnormalities.
This case comes from Function and Genetics in Dystrophin and Dystrophin-Related Proteins in Muscle by Matos et al. This graphic shows eutrophin in normal and dystrophic muscle. Immunohistochemistry was used to visualize dystrophin, shown in the top row, and eutrophin, shown in the bottom row, in human muscle taken from healthy controls, which is shown in the first column, patients with Becker's, shown in the middle column, and Duchenne's muscular dystrophy, shown in the third column. Here, the results show that defined borders in the muscle of the normal muscle compared to the Becker's muscle with little outline, and muscular dystrophy with no border. These results show the differences in proteins found in normal and diseased cells.
IHC can be used in other ways from the immunofluorescence method. This can include the enzymatic method, which identifies antigens of interest by exploiting the principle of antibodies binding specifically to antigens, which ultimately uses enzymes to label the antibodies. This method has many advantages because a fluorescent microscope does not have to be used, and the stained samples can last a very long time. There are two procedural techniques that can be used when performing the enzymatic method, and they are simply defined as direct, which is a one-step procedure method, and indirect, which is a two-step procedure method. But both procedure methods will acquire the same general results. Another way IHC can be used is seen in the affinity method, which is generally used to amplify the target antigen signal. There are two widely known affinity methods. One is the ABC method, also known as the avidin biotin peroxidase complex, and the other method is the labeled streptavidin binding, or LSB. Both methods have their own separate ways of amplifying the target antigen signal, but in the end, that is exactly what both of these sub-techniques are trying to do.
Recent advancements in immunohistochemistry include the ability to better differentiate between tumor types and the use of biomarker detection in clinical practice. Studies and experiments have been performed using IHC in order to detect presence, morphologies, and genetic characteristics of different tumors. Protein expression through IHC testing is critical in monitoring the progression of cancer and helps with treatment. These advancements help lead to more personalized treatment in diseases and cancers. Here, myofibroblasts and prostate cancer can be seen. These images show the different biomarkers that a tumor contains. This is an example of how immunohistochemistry recently advanced to allow for more information when analyzing samples.
Here we're excited and thanks for learning about immunohistochemistry.